Hello, and welcome to this celebration of the iGEM team of 2025. My name is Roger. I'm the head of Department of Biosciences, and the iGEM course, I would say, or competition is a collaboration between the Department of Bioscience and the Science Library that has been going on since 2014. So this has a long-standing tradition. This year, our team actually managed to win the gold medal, which is a great achievement, and was also nominated for 2 other prizes that you unfortunately didn't win. But we are here today to celebrate that gold medal, and I think I'm going to let the iGEM team introduce themselves. So, come on.
This work?
Oh yeah.
Sorry. Yeah. You can start. Go ahead.
Hi, welcome everyone. We're here to celebrate our gold medals and the 2 nominations, just in case. This has been a very exciting journey for all of us. Of course, there's been a lot of challenges as well. But I'm going to start with introducing what iGEM is, just very briefly. So it's an international genetically engineered machine. It's the world's biggest competition in synthetic biology. And over 400 teams from different academic institutions like universities or high schools, they participate. And Grand Jamboree is usually in Paris. There's so many opportunities by participating in iGEM, and we have managed to mention some of them here. It's not only for us. I mean, like I'm just gonna start by like our team experience. It's not just about like a competition. It's about making friendships. It's about making connections. It's about engaging with fellow students when you go to, for example, when you attend the Jamboree in Paris. You meet so many new people. You get so many inspirations from them, and at the beginning, you get to, before you make a potential iGEM team for your university, you get to pitch different projects. Different students come, join the meeting, and they pitch their projects, and then we vote for one of them as the eventual project. There's so many opportunities. You get to work in the lab. You get to work outside the lab. Depends what your specialty is. For our group, some of our students were not biology students, so they also got to experience how the whole lab work thing works. They saw like so many instruments in the lab that they weren't familiar with, and it was very fun. You also get to travel abroad to different conferences or meetups. It could be a Scandinavian meetup. It could be a European meetup. This year, we were so lucky to go to 2 of them. One of them was in Frankfurt. The other one was like a Nordic meetup in Denmark. It was very fun. We got to connect with so many different students from different countries and exchange ideas. And you also have to prepare for the final competition, which is very challenging because you have a lot of work to do, and you might not feel that it's so hard, but you will feel it towards the end of it. And yes, I just remembered to mention that we were so lucky by attending the Nordic meetup. We won the Golden Pipette, so that means that the next Nordic meetup is going to be here, held in Oslo. It's very exciting. In addition, you also get 10 points for the whole course, which can be, this whole course is during 2 semesters. So in summer you just have to work hard and do all the work for the lab and additional wiki page and some more work. And yes, as mentioned, we have won the gold medal and we got 2 nominations for the best wiki page for our teammates' efforts in making the whole wiki page and the contents, and also the best part, the biological part. And this is the medal that I'm wearing. Very exciting, guys.
So you might be wondering, well, how did we win the medal? And that is through our project called LactlQ. Now, as you all know, we are in Norway. And in Norway, there's a lot of athletes. We are quite an athletic country in general. So for our project, we combined both sports science and synthetic biology to create a real solution to world problems, because that's what iGEM is about.
Felix, one thing, I guess, like we are presenting ourselves, and then I think
Yeah.
we do the presentation afterwards.
So do you want to...
Right?
Are you doing the presentation now, or are you presenting yourselves?
We can do it either way.
All right. Yeah, use the what you have here.
Okay.
But do we want to do that team slide? We have a team slide.
Yeah, we can do the introduction of the team slide. And then we can do the entire presentation. I don't know.
Okay, so do you want to do this one first?
I guess. Yeah.
Yeah.
we can do it.
Okay. So, yeah, to introduce us, each one, or yeah. To introduce ourselves, we are a team of both people from biology and entrepreneurship. So my name is Felix. I have a bachelor's degree in biology from Kristiansand, and I moved here to Oslo to study my master's in entrepreneurship and innovation management. And do you guys want to hear them? Yeah.
I'm Furu, and I have my bachelor's degree in general biology, and I'm doing my master's degree in toxicology and environmental science.
I am Khayal, and yeah, I have my bachelor's in my bachelor's in instrumentation and control, and yeah, I come from India, and yeah, I have some experience from previous jobs.
This doesn't seem to be working.
My name is Alan, and my background is in genetics, and I'm a cell biology student.
We have the extended team there.
Peder is my name. I have a background in information science and entrepreneurship and innovation management.
Hello, I'm Elina. I have a master's in molecular biology and biochem.
Hi, my name is Amanda. I have a background in sustainability, and I'm also doing the master's in entrepreneurship.
My name is Jon Einar. I have a background from mechanical engineering, and I'm also doing my master's in entrepreneurship.
Hello, my name is Diego. I'm working in the modeling, and my background is in genetics, and I'm studying a master in evolution.
And here we have our supervisors. They are going to introduce themselves.
Okay, my name is Dirk Link. I'm a professor in molecular microbiology, and I've been involved in supervising iGEM since 2014.
And I am Athanasius, a researcher in group of Dirk, and I have been involved in that since 2015, more or less, 2016.
And I'm Kirsten Bosch Haraldsen. I am a first academic librarian and in biosciences support. And I've been with iGEM facilitating for all these years, from 2014.
Thank you very much. We've been so lucky. I've been working with these amazing people.
No. Okay, we can start with the presentation then.
Now?
Yeah.
Yeah. So to introduce the problem we have been working on, currently in a lot of sports, professional athletes take lactate tests in order to get to know their intensity. So the way it works is you poke your finger with a needle, you extract the blood on a strip, which you put into a device. And that's how nowadays most professional athletes train to measure their intensity in order to not overtrain and overstretch their sort of internal battery, you could say, right? And lactate is a good metric because it is the direct product of going from aerobic to anaerobic. So it's very much scientifically measurable, and unlike other methods, it's not that much affected by outside factors such as temperature and your sort of blood sugar and other things, which means that it's generally quite reliable. Now, as I mentioned, nowadays, do you have the problem of inconvenience, the fact that you have to poke a hole in your finger and then you will bleed? You also have the fact that you can only do these tests between each running interval. So you'll do a run, and then you'll poke the test. It's kind of like if you were to drive a car, you could only look at your speedometer after each driving session, which doesn't really make sense, right? You would preferably be able to know the speed while you're driving and not afterwards. So all of these problems accumulate in the fact that you also have to purchase these devices and needles and strips one by one, which leads to both bio-waste and high recurrent costs. And these are all problems which centralize around how athletes can train effectively and also the economic waste around it. Now, we got this problem from Amanda, who is the current 800 meters national champion here in Norway, and she knows a lot about the problem from the athlete community. So through her resources with both athletes as well as other medical professionals, we have sort of interviewed and reiterated the problem to know the real sort of causes behind the problem and what the athletes need are in order to know what kind of solution we should aim for, which is what we're going to introduce next.
Yeah, so we came across a lot of research, and we found that the best way to approach this issue is by utilizing microneedles. Once we figured that out, then we realized that the microneedles needs to be coated with some enzymes, and we needed some experts to do that part, and that's where we reached out to the other biology students. Anyhow, with time we figured it out that there are two different enzymes, lactate dehydrogenase and lactate oxidase, which reacts with the lactate molecule in your body and produces some sort of signal. And then we had to capture that signal and then kind of amplify and send it to your watch through Bluetooth. So that was the basic idea. And just a heads up that the microneedles, they look big on the screen, but they are only, as you can see, it's just 1 millimeter big. So it just goes below the dermal layer.
So the main component that we needed was the enzyme that we could react with lactate, and we basically have the molecular flow here. We transformed the bacteria with the gene of interest. So we had two enzymes, so in parallel we ran 2 experiments, and then we isolated the plasmids, and then we transformed another strain of bacteria with those plasmids, and then we run some functional assays to see that our enzymes are working. And then we had large-scale protein production, and then we performed the electrochemical assays to see if we can detect it. Yep. So I thought Diogo is going to be here. Mm-hmm. We had the NADH consumption, so basically through the detection of that NADH we could see if the assays could work. And here you can see that based on the concentration of the NADH, we could detect the enzyme.
Yeah, the enzymes were functional.
Yeah.
Yeah, for the model, we start with this classic model for Solomian and front. This is a model with 2 spaces, and this model should be able to predict the lactate levels. It's a lactate genetic model. And we start with this one. This is a 4-year-old model, and those are the results by using just that model. As you can see, the target curve and the simulations, they are really close. So the idea is this model is really good, but we need to improve it because this model is only able to predict lactate after resting, and we want to predict lactate while we are exercising. So we introduced 4 different equations to moderate, to change the saturation in the radiation rate, and by doing that, we were able to predict the behavior of lactate while doing exercise. And this is the first time this would be someone do this, so this is a really good result.
And how to pack it all into a product. So yeah, we started designing the product, and then this is the final elements that we have kind of narrowed down into. It starts with the microneedles, which are being coated with the enzymes. They are packed in a casing, and which has a PCB, and through which it detects the signal and sends it to the chip. And it's a Nordic semiconductor chip, which basically processes this data and has a Bluetooth module inbuilt and can transfer the data via Bluetooth to the watch or to your phone. And along with that, it has a battery component to support it for a longer time, and an adhesive so that it sticks to the athlete and doesn't fall off with time.
Now, the hardware development that Kyle just talked about is obviously something that's going to take a lot of time, because contrary to, for example, developing an app, making hardware requires you to talk with both manufacturing parties, securing IP, and knowing exactly what components you need to order, which is exactly why we have made a plan for how we're going to continue with this progress. And as you can see on the left side, all of the points marked with orange are our achievements in iGEM. So that's mainly the results that we managed to prove in the lab for establishing a proof of concept. And then looking further into the future, we'll just build the device that we just mentioned.
Yeah, and you might be wondering, okay, who wants to measure lactate, right? No one's interested in doing this, maybe 10 athletes. So yeah, to answer that question, we have looked into the market size, and it's a pretty huge market because it's not only runners who are using it. There are a lot of people cross-country skiers, even in swimming, rowing, and also the latest discovery is people are measuring lactate for horses before they race them. So yeah, it's a pretty huge market. And to quantify exactly numbers, it's 9.58 billion euros as a total market in Europe. But if you narrow down to Norway, it's 145 million euros. And if you just consider just 10% of that market, which should be our SOM, which is the serviceable operable market, that's something which you can practically reach, or something what is not too idealistic, that is 14 million euros. That's just the sports-related market. Apart from that, if we get proper certifications and accuracy levels, we can move into a medical market as well in the future.
Now, of course, none of this progress would have been possible without our wonderful partners. So we have, obviously, here at UiO, the Evogen specifically for providing both access to the laboratory and excellent advice from Athanasius, especially want to thank for helping us progress in the lab as well as Startup Lab for getting a lot of these connections for the electronics part. So developing the hardware is obviously something that none of us had experience in. So Startup Lab has been very helpful for connecting us to, for example, the physics department, where we managed to include PhD students and professors to also work on our project. So this project has been very interdisciplinary, as well as Inch for funding us as a student startup. And speaking of funding, we have also gotten quite a few sponsors, or rather, the sponsors that we have been very helpful, because getting sponsorships in Norway for biotech, as some of you might know, is quite difficult. So we want to thank both Digital Life of Norway, Avantor, as well as Sorchim, which is one Swiss company, which provided us with some of the lactate oxidase samples, which are otherwise very expensive to get on the market, as well as Co-Create, which is in collaboration with Inch to help fund students who want to do their own projects. And the team, which, yeah.
Yep.
Thank you.
Thank you. Sure. Yeah, if any of you have any questions, or yeah.
You were talking about the wiki and the nomination for best part. Could you just tell us what it is? And the best part, what is that in iGEM?
Then you want to take this?
Yeah. So other than the competition over the medals, which are gold medal, bronze, and silver, there are some other nominations and winning some of the other parts of the competition. So there are several of them, including Best Wiki, which is the platform. Basically, the teams put every information on that. It's some biological part, the inserts and the the genetic parts that are you introducing for the first time there, and also the marketing part and etc. Basically, we were nominated for 2 of those parts. One is the Best Wiki, the platform that I mentioned, and the other one was the best basic biological part. Yeah.
Would you mind like a little bit details on what is the basic part?
So the proteins or the enzymes that we engineered and used it for those reactions were two. So for those two, we had two inserts, meaning that the gene sequences that we designed vectors for, and basically we put all of the information in that platform, and we won for that. We didn't win it, we were nominated for that. Yeah, so it's the best basic biological part.
Yep.
So what is the actually criteria in part of doing innovative project in the lab? Is there any criteria else to get a medal as gold? What did you do to get the gold? What is the criteria to get it? Because it's not only you that had that golden medal, so it's a lot of work to deliver.
I think Felix, you want to take it? Yeah.
So the medal itself is about the project as a holistic structure. So, for example, there's requirements for bronze, silver, and gold, and then if you excel on any specific part of your project, so let's say, for example, you have made a biological part which is sort of above the average of what you can expect from our program, or if you have made a website that has a quality that's above the average expected, then you'll get the nominations in addition to the gold medal. So the medals are sort of a baseline, and excellence in any specific fields are what those nominations are for.
So you are exposed to some judging session that you have to stand up for your work.
Yes.
In Paris.
Yes.
So every team has like a session where they will go into a room with judges, and they will evaluate your project and give questions to it. And obviously they will have read up on your project beforehand. So the session is about questioning and sort of making sure that the team has control of their own project, and in a sense that they have developed it organically and know that they're what they're talking about.
Nice.
Okay. There are questions here.
Hello, very nice presentation, and I love the idea and I love the product.
Thank you.
So, having seen what you have done so far, I'm very curious to see where your plans are for the future. For instance, where do you see yourself in 5 years from now? And what do you have to do to get to that place you want to be in 5 years from now?
Yeah, there are a lot of things for this moving machine. So one of the first thing is whenever you want to build a product, people ask you the first question, is the proof of concept done? That basically means that, is it functional? Does it work? Like, can you show me in the lab if this works or not? And that's like the first situation that we have to jump through, because as we have shown that we have developed the enzyme, now the enzyme needs to show that it reacts with the lactate and produces the signal which you can kind of quantify and document. So that would be like the first step, which is showing the proof of concept that, okay, conceptually this works. Then you take it further and then kind of introduce different sort of variances. For example, now we are testing just with lactate, but what if there is some noise? What if it is actual interstitial fluid where you have glucose and other enzymes, other things available as well? And would you be able to measure in that situation? Once these questions are answered, then you go for a big model, which people call as MVP, minimum viable product. And that's like, if you are assuming the patch to be this small, this version would be like massive. And you just show that, okay, electronically it is possible. You did something in the lab, now you can do this at the electronic level. Then you go for approvals and regulations. Once they are cleared, then you start making it on a smaller scale. And yeah, so it has a lot of steps. And what from an entrepreneurship perspective, you can say as de-risking. So each thing is a risk, like would you be able to do that? And then you just reduce the risk for people who are kind of investing in you, supporting you, are part of the team, and they believe more in the idea, and it becomes more real. So that's the plan.
I wonder, you could put up the wiki here just to show the product. Is that possible?
Want to do that?
But at least you did have some help with the. You need to also give a video during the competition.
Yeah.
And take a lot of pictures,
Yes.
as you had here.
Project or
So after the streaming, we could maybe show the promotion video. Is that
Sure.
possible?
Yeah. I think we can
And
pull it
I
up.
would also say thank you to Simon at the Science Library that has helped a lot with all that delivering for the competition.
Yes, a lot of cinematic shots.
Yes, without you, we would never have such amazing videos and portraits. Thank you.
Right.
Okay.
Should we get off
So
the
then
stage?
we can
Huh?
say thank you.
Yeah. Huh?
For the, thank you so much.
Yes.
Or did you have anything more to?
No, we are happy.
Okay. And the supervisors. Well, it is a very nice presentation. Thank you so much.
Welcome.
And you did a great project, really.
Yes.
It's very interesting. And it, as you say, it is very interdisciplinary group of young scientists, actually. Mm.
Thank you so much.
So thank you for also bløtkaken.
Yes.
Yeah.
Yes, definitely.
Yep.
Thank you all.
I don't know.